Welcome to the next lecture. This lecture is about cell compartments. As always, let's recap what we discussed in the previous lecture. In the previous lecture, we talked about membranes, and we talked about osmosis and diffusion. We said that membranes are semi-permeable, they're sort of fluid, and they can contain proteins. We talked about cholesterol, which allows the membrane to stay fluid enough not to be too fluid, and not to be too rigid in changing temperatures. We discussed diffusion as it occurs across a membrane along the concentration gradient of the solute in question. We said that solutes move always from a higher to a lower concentration. We said that that can also be the case for water, which can also move from a higher to lower concentration. And we said that membranes can restrict solute passage, but membrane proteins that can be integral to these membranes can facilitate passage of specific molecules. And then we said that membrane proteins can allow passive diffusion or active transport. And active transport requires ATP, but passive diffusion does not, and it can be facilitated through proteins, but it would still be passive diffusion. It would still be along a concentration gradient. And then we said that if a membrane restricts solute passage, water will move through the membrane to equilibrate the concentration difference. And that is the principle of osmosis. So today, we're going to look at a number of membranes as they are found in cells in various compartments, and as they shape the various compartments, and as they basically produce these various compartments, because without a membrane around them, there is really no compartment. So let's first take a look at prokaryotes versus eukaryotes and just sort of see what the difference in their cells is. So the prokaryotic cells are much smaller than eukaryotic cells. That's already one thing. And prokaryotes, of course, are, for example, bacteria. So bacteria are prokaryotes. They are very small compared to the much larger eukaryotes. And prokaryotes, they have DNA, but the DNA is not contained in a nucleus. So the nucleus is what makes the eukaryote proper. Nucleus is what eukaryote actually means, translated from Greek. And prokaryote is sort of a prototype of a nucleus. That makes the difference. So in prokaryotes, the DNA is concentrated in a discrete area of the cell, in a specific area of the cell. It's not just sort of floating around, but it's not bounded by a membrane. That's why it's called a prokaryote. And then eukaryotes have a membrane-bounded nucleus, and they're much larger. All right, so let's take a look and see what eukaryotic cells look like from the inside. And we want to make sure that we distinguish between plant cells and animal cells. Plant cells have a few features that animal cells do not have, and I want to point those out in the next slide. But let's first look at animal cells, and let's look at cells, at the contents of cells that all cells, both plants and animals, have. So here's an example of an animal cell, and you see that it has a nucleus. So inside, that is the nucleus here. It has relatively large organelles that are bound by a membrane that are called mitochondria. And mitochondria are those places in the cell where ATP is produced or energy is produced. And then we have the cytoskeleton. Those are fibers that are made up of molecules that sort of string throughout the cell. And they can give the cell some sort of structure, but they can also be used as a highway, if you will, for other proteins to traffic along them and rapidly move molecules from one place to another place in the cell. So that is what the cytoskeleton is also used for. Then we have peroxisomes. Peroxisomes are small organelles that break down fats. Then we have the endoplasmic reticulum. And the endoplasmic reticulum is a membrane system. It's quite a large system, really, and it's large, but it's made up of these very thin membranes, and so it's pretty much not visible in the light microscope, despite its size. So the endoplasmic reticulum is involved in protein and in lipid synthesis. And then we have the Golgi apparatus, which is sort of an extension of the ER, or endoplasmic reticulum, but it's separate. So it's not connected to the ER and therefore not connected to the membrane that surrounds the nucleus. The Golgi apparatus, or the Golgi, modifies and shuttles proteins around the cell. And we'll take a closer look at that in a little bit. And then we have the lysosomes, which break down these macromolecules and then recycle the content. So all of those are also found in plant cells. But plant cells have a few extra features that are not found in animal cells. First of all, plants have cell walls. We discussed this already when we talked about osmosis, when we said that plants that are that live in fresh water and they have salt inside, they have solutes inside, they benefit from having a cell wall, because water that keeps streaming in to dilute the inside of the cell would expand the membrane maybe to a bursting point, to lysis. And so the cell wall prevents that from happening because it presses against the membrane from the outside. Plant cells, of course, also have chloroplasts, and chloroplasts themselves have membranes inside. We'll take a closer look at that at the end of the lecture. That is the site of photosynthesis, of course. Then plant cells have plasmodesmata. Those are little passageways, if you will, that connect two plant cells with each other, that otherwise would have a hard time connecting because of that thick cell wall that's around them. So plasmodesmata are basically membrane holes that are lined with membranes. And the membrane is the cytoplasmic membrane here that goes through this hole and then connects to the next cell. And that really makes cells connected in plants into one large body of cells, if you will. So that helps plants for cell-to-cell communication. And then another thing that's found in plants that's not found in animals are the vacuoles. And there's a central vacuole, and there are also some smaller vacuoles inside a plant cell. Vacuoles are filled with liquid, and so they can contain they can maintain the turgor pressure, as it's called, the pressure that's inside the cell that's sort of pressing outwards to give the cell its ability to stand up if it doesn't have a skeleton or a woody stem. So that filling this central vacuole with liquid allows the plant to be turgid, to be upright. It can also serve as what some people call the trash can of the cell. So plants can sequester molecules into the vacuole that are either harmful or that are no longer or currently not needed. And so they can sort of be placed there for now. All right, let's take a look at the individual parts of the cells. Let's start with what's called the endomembrane system. So this is an endo, meaning inside. Membrane, of course, is a membrane. And the system means that it's a whole set of membranes that are sort of crisscrossing through the cell, that are somewhat connected in some places, but in other places only connected via docking of vesicles. So what do I mean here? So we have the nucleus, and we have the nuclear envelope. So that's the membrane that's around the nucleus. And it extends into the endoplasmic reticulum. So you can see that the membrane here sort of folds outwards and produces this membrane system that then has all these little folds and crannies and spaces. You can also see that the endoplasmic reticulum comes in two flavors, if you will. There's the kind that's called rough endoplasmic reticulum that has these yellow dots on here in this graphic. Those are ribosomes. We'll come back to ribosomes, but they're studded with ribosomes. And then there's this smooth endoplasmic reticulum that does not have that. And you see that the Golgi apparatus is connected, but not directly, with the ER. It's only connected with the ER by the virtue of the fact that these membrane folds can sort of bud off and then flow through the cell and dock onto the Golgi apparatus. And the Golgi apparatus has all these membrane folds again, and they can bud off, or they can fuse with the Golgi. And so through that system, compounds that are inside the ER can be placed into a vesicle, and then that vesicle can dock onto the Golgi, and then from there, things can happen inside the Golgi apparatus that we'll discuss in a second. And then from there, these membranes can bud off again, dock with a membrane, and then the outside membrane. And then from here, the molecules can either be excreted or they can be stuck in the membrane and become part of the cell membrane. So that's why it's called an endomembrane system. So general functions you could summarize by saying it shuttles molecules through or out of the cell without the need to cross a membrane, because they're always inside these membrane-bound vesicles. It creates an internal compartment that's separate from the cytoplasm. Both of those, of course, are inside the cell, but there's another internal layer, if you will, inside the cell with the help of this endomembrane system. And you can use it for excretion, but also for picking up of molecules. That's called exocytosis or endocytosis. All right, so the nucleus, of course, contains the DNA. And the nucleus, as we just discussed, has a membrane that is connected to the ER. And then in this micrograph, this is an electron micrograph. So it's a specific type of microscope with which you can focus onto very, very small things that you cannot see in a light microscope. So this here is only 1 micrometer. So it's a thousandth of a millimeter, which is a thousandth of a meter. So very, very small. Contains the DNA. That's the place, of course, where the RNA is synthesized inside the nucleus. It's surrounded by the nuclear envelope. That's the membrane around the nucleus. And these pores allow the passage of RNA from the nucleus out into the cytoplasm, or the import of certain proteins from the cytoplasm into the nucleus. Okay, so we already talked about the ER a little bit, but here are some pictures that I want to show you that are either of the rough ER, so that's the one that's associated with ribosomes, or the smooth ER that lacks the ribosomes. So smooth ER up here, rough ER here. And so the rough ER that contains the ribosomes, that is using the ribosomes to produce proteins sort of in place where they are. So they're studded onto the membrane, but as you can see in this little close-up, the RNA is threaded through the ribosome, and the ribosome is bound to the ER. And as it's making that protein is placed inside the lumen here of the ER, so the inside of the ER. And so therefore, it's shielded from the aqueous hydrophilic cytosol or cytoplasm, and it can then stay inside these vesicles that can then dock onto the Golgi and eventually be excreted. But it can also be modified inside while it's in there. So much of that modification happens in the Golgi apparatus. So here is still our ER. There are some proteins maybe inside this vesicle that's now docking onto the Golgi apparatus. And here's a close-up of the Golgi apparatus. You can see these little parts here that are going to bud off, and these flatter regions that are the membranes inside of which are the proteins. And so what happens in the Golgi is that it modifies the proteins and it sorts the proteins. It decides where these proteins are going to be sent within the cell. And during the modification process of proteins, certain things can be added, subtracted, modified. So molecules can be added onto that protein. One group of molecules that frequently added to proteins are sugar molecules. Those are called then glycoproteins. And then the Golgi moves these proteins and the lipids from the ER to the outside, for example, here through exocytosis. Okay, so then we have lysosomes. Lysing means to break down, and somes, of course, means little bodies. So lysosomes are breakdown bodies. They are surrounded by a membrane, and they are filled with enzymes that can break down large molecules, macromolecules. So in other words, proteins, sugars, fats, even nucleic acids. These lysosomes are able to do that while the rest of the cell is not able to do that because of these enzymes inside, but also because there's a certain pH, so that's at the acidity level within the cell, that is lower in the lysosomes than it is outside in the cytosol. So if the acidity was as strong in the cytosol, cytoplasm, cytosol, I sometimes just use these two words interchangeably. If it was acidic in the cytosol, then other things would get destroyed as well. And so you don't want that to happen. So you just want to destroy certain things at a given time. And so therefore they get imported into the lysosomes where the pH is lower, where certain enzymes are active, and where they then break down these molecules and then transport them out. And so you can see that this pH change occurs because of proton pumps. We talked about membrane proteins in the previous lecture. We said that protons can be pumped through a membrane. So this is such a proton pump. This is not so much in order to build up or maintain an electrochemical gradient, but it is for simply lowering the pH. So more hydrogen ions means lower pH. The greater concentration of hydrogen ions, the more acidic and the lower the pH. So that happens in lysosomes. All right, so that's all part of this inner membrane system. These are all somewhat connected. Even the lysosomes, they can bud off from the membrane systems. I said that proteins get sorted, and I might better say what I mean with sorted a little more specifically. When I say sorted, I mean, where are they going to go within the cell? How does the cell know where a protein should go? Should it go into the membrane? Should it be excreted? Should it maybe be excreted into the cytosol rather than on the outside of the cell? What is the function of that protein? And so in order for these proteins to know where to go, if you will, they get little signaling sequences. And so a sequence, of course, is the certain order of amino acids within a protein. That's what we call the sequence, right? That's the primary structure of a protein, is the amino acid sequence. And so part of that amino acid sequence can be certain amino acids that in the combination that they have together signal where that protein is going to be delivered in the cell. So it tells other parts of the cell that this protein, because of this particular signaling region, has to be delivered to the outside, or maybe to the nucleus, or maybe to other parts of the cell. And so that's why they're called signaling peptides. So in this case, we have no signal peptide. Here we have an amino terminal signal. Remember that there's an N terminus and a C terminus in every protein. That's where you have the amino group or the free carboxyl end. We talked about that at the very beginning when we discussed the structure of proteins and how we make peptide bonds and how that's always requiring the formation of a peptide bond through a condensation reaction of an amino group and a carboxyl group. So those that have not been reacted, they're leftover ones, if you will, on the outside of this chain. That's the amino terminal end or the carboxyl terminal end. And at the amino terminal end, we can have certain signals that signal the cell machinery to target these particular proteins either to the chloroplast or to the mitochondrion. And then we have certain internal signals. These are signals that can bind to other molecules that then can bind to the outside of the pore of the nucleus, and they can then import these proteins back into the nucleus. Certain proteins, of course, are required for the nuclear activities in order to do transcription, for example. And all these proteins need to get into the cell somehow, into the nucleus. Most of them are translated in the cytosol. So how do they get back into the nucleus where they're working, where they're acting in the transcription process? So they have to be targeted back through the pore into the nucleus. And that's what these signaling sequences are for. So I write here signaling sequences are sort of like zip codes. So there's a certain number of amino acids stuck together in a certain way, just like a zip code, 98416, right? It's a certain combination of numbers that together gives a code, and that tells the postal office where to send a package. Or in this case, it tells the cell where to send this protein within the cell. Okay, so let's quickly look at secretion versus retention in the membrane. So we talked about membrane proteins, and we said that they're integral membrane proteins. They have to get into the membrane. And we also said that it was kind of hard for these proteins to get into the membranes if they have if they're particularly hydrophobic, then it's difficult to get through the hydrophilic head. If they're hydrophilic, then it's difficult to get through the hydrophobic tail part of those phospholipids that make up the membrane. So how do they get in there? How do they get stuck through the membrane? Well, they can get stuck through the membrane early on, very early on, as they are being made, essentially. So look at this picture up here where you have the RNA threaded through the ribosome that is stuck on the ER, in the rough ER, so that's the place where the ribosomes are. And as the protein is being made, it already goes through this membrane here, right? So it could be, it could get stuck, if you will, in the membrane. And then if it's stuck in the membrane, it can just stay stuck in the membrane. And then as being part of the membrane, it becomes part of a little vesicle that buds off from the ER, docks onto the Golgi, still staying in the membrane, and then it gets changed, it gets maybe glycosylated, modified, right, within the Golgi, and then sorted, and then the next vesicle buds off. And if this buds off and fuses with the plasma membrane, you see that the protein is still stuck in the membrane, and it's still stuck in the membrane as it's fusing with the membrane, and this vesicle becomes now part of the membrane, the cell, the plasma membrane. And so the protein still is stuck in the membrane, just where it was synthesized. And so that way you can easily insert proteins into membranes and then just sort of keep them in there because they were synthesized while already stuck, or as they are being synthesized, they're stuck in the membrane. And then that allows the transfer of this piece of membrane really to the outside. But there can also be the secretory system where you have secretion of certain compounds. Cells have to secrete certain things, right? Hormones get secreted, they get made, they get secreted, other substances get secreted. And so they have to come out of the cell somehow. And if they are large and maybe there's no transport, protein transport, membrane transporter, then they can be synthesized, kept inside the endomembrane system through the Golgi apparatus, and then as a vesicle through the cytosol, and then this membrane will dock on here, fuse with the outer membrane, become part of the outer membrane, and then expel its content into the extracellular fluid. So I say here the ER together with the Golgi allow proteins with different properties, whether they're hydrophobic or hydrophilic, for example, to enter the membrane and or to avoid the membrane altogether. So they don't have to go through a membrane this way if they're too big, for example. All right, so most of that was part of the endomembrane system, different organelles, if you want to call them that, or membrane compartments, as your book calls it. But there are two very specific organelles that are not part of the endomembrane system, and that's the mitochondria and the chloroplasts. So let's take it the mitochondria. The mitochondria are also bounded by a double membrane, so there are two layers of lipid bilayer membranes. And inside the mitochondrion, this is an EM electron micrograph of a mitochondrion. Inside, along these membranes, ATP is produced. And exactly how that is produced, we will discuss in great detail coming up in the next few chapters. But this is where it is produced. Mitochondria are big enough, and they are also dense enough to be seen in the light microscope. So if you have looked at cells under the light microscope, human cells, sometimes you can see the mitochondria in there. So the function of mitochondria is to produce most of the cell's energy in the form of chemical ATP. And exactly what ATP is, we haven't discussed yet, but we will. It's a chemical at this point, that's all we need to know, that carries in its bonds a high amount of energy. And that is being produced here in the mitochondria. All right, and then the last one that we want to discuss are the chloroplasts. Of course, chloroplasts are only in plants or in green algae here. Chloroplasts also contain, bounded by a double layer, a double membrane, each with a double layer, right? So each membrane is a double layer membrane, but there are two membranes surrounding the inside of the chloroplast. And then inside the chloroplast is a third set of membranes. And those are called the thylakoids. The thylakoid membrane system is inside here of the chloroplast. And along those thylakoid membranes, that's where much of the ATP production in plant cells occurs during photosynthesis. We'll also spend a lot of time later on discussing how exactly that works. All right, so that's the chloroplasts. So then there's just the summary. And so for the summary, we have talked about prokaryotes versus eukaryotes. In this lecture, we said that they are distinguished by the lack of a proper nucleus in the prokaryotes, and the larger cell size of eukaryotic cells, and the fact that eukaryotes have membrane-bounded nuclei, while prokaryotes don't have that. And then we talked about the cell compartments in eukaryotic cells. We said that they contain an endomembrane system, which consists of the nuclear envelope, the ER, the Golgi vesicles, and the lysosomes. We said there were mitochondria and chloroplasts that are not part of the endomembrane system. And we said that the chloroplasts and the central vacuoles are only found in plants. And of course, we discussed what the functions of each of these organelles and membrane systems are. We talked a little bit about the modification of proteins. Glycosylation is one thing that I suggested would happen in the Golgi apparatus. And we talked about the transport of these proteins throughout the cell to their final destination, which can be inside the cell, can be inside the membrane of the cell, or it can be outside the cell, as is the case with secretory proteins. And so for the cell to know where these proteins have to go, they have signal peptides, and I likened them to zip codes. So that is our lecture for today. I will see you in class.